All right, guys, here we are. Topic of the slideshow is RNA, ribonucleic acid. So one of the things that you want to know at the end is how RNA differs from DNA. And then how does a cell make RNA? Okay, so let's look at RNA and DNA. RNA, which is called ribonucleic acid, and DNA is deoxyribonucleic acid. Ribose is a sugar. That is that in a nucleotide, you have the sugar piece. The nucleotide is the A, C, T, G, and now we're going to have a U. That uracil has ribose as the nucleic acid. So the sugar in RNA is ribose instead of deoxyribose. All right. RNA is generally single-stranded, not double-stranded. So DNA is generally double-stranded. RNA contains uracil in place of thymine, thymine. So instead of a T, it has a U. So A doesn't pair with T, A pairs with U. So if you see the letter U in a single-stranded molecule, more than likely it's RNA. So we have three types of RNA: messenger RNA, ribosomal RNA, and transfer RNA. The name says what it does. All right. So an mRNA is how we denote messenger RNA. An mRNA molecule is a copy of the portion of the DNA that's going to be used to make a protein. So mRNA is made in the nucleus, and then it travels out to the cytoplasm, to the site of protein synthesis. Ribosomal RNA is where ribosomes are made up of ribosomal RNA, and that's where protein synthesis occurs. Protein synthesis is to synthesize, is to put together. And so the proteins are put together at the ribosomes. And those are made up of two parts. So you got two parts, ribosomal subunits that fit together that make up a ribosome. And they consist of several molecules of RNA and as many as 80 different proteins. Transfer RNA. So transfer RNA, during protein synthesis, that transfer RNA brings the amino acids from the cytoplasm to the site of protein building, protein synthesis, and that is a lock and key kind of mechanism. So we're going to take a look at the process of protein building through how that happens. And so we start out with the DNA, and the DNA is the code for everything. All right. So the DNA codes for a protein. Now to get the protein, to get the code out of the nucleus, we're going to use messenger RNA. So the RNA polymerase is going to assemble that strand of RNAs and use the DNA here as a template to do that. And so we have adenine, cytosine, guanine, and uracil. Okay. So those are the different pairs. So if you notice the top one here is a G, and that's pairing with a C, a cytosine. And then we have a thymine, which is in DNA. And then that pairs with an adenine, the green one. Well, the adenine here, now because this is RNA, it's pairing with uracil, right down here. So the base pairing rules switch slightly with RNA to DNA. There we go. All right, so when RNA is being synthesized, there's these pieces in there that are called promoters. And so this RNA polymerase binds to these regions of DNA that have specific base sequences, and those are called promoters. So the question would could be or is, how does the RNA, where does the RNA start? Where does the RNA start to replicate? Okay. And it's going to look for a sequence, and the RNA polymerase is going to bind to that site right here, that there's what's called promoter region. Promoter, right? To promote, to move forward. So that's where the DNA is going to be read at a promoter region. Now, there's a couple of other parts. So once we pull this strand of RNA off, there's a portion of it, it's going to basically be edited. And there's some parts in there that we're going to edit out. So there's introns and extrons. So the introns are the part here that's going to be removed. So we copied this whole strand of messenger RNA. There's introns and exons, and the introns are going to be taken out. The exons are spliced together, and that becomes a piece of messenger RNA. Okay, so there's our introns, and there's our exons, and the exons are going to be ultimately the components, the parts of the mRNA that are going to go out. All right, and there's a cap and a tail, so you got to know when to start reading it and when to be done reading it. Okay, so that's that's the introduction to RNA. RNA is an exciting, it is really the deal, right? Because the DNA holds all the code, but in order to get the code out, you have to have RNA. So messenger RNA is a big deal. And messenger RNA is now being used to produce some of these vaccines we're hearing about, in that it's producing a protein, what's called a protein epitope, or a part of a protein that exists on things that we want to become immune to. And so the RNA then is being, we get injected with this messenger RNA molecule, and then the messenger RNA molecule makes these proteins that are recognized as not part of our body. Our immune system responds to those, develops antibodies to them, and we basically have a vaccine for a virus. So that is it for now. I'll